The next series of experiments will show the effects of bacterial targeting of metastasis in mouse models. In this model, the tumor cells were injected into the inguinal lymph node, where they trafficked to the axillary lymph node and grew. The bacteria were then injected into the inguinal lymph node and then trafficked to the tumor in the axillary lymph node and could cure the tumor. This is a movie of the bacteria entering the axillary lymph node growing tumor when injected into the inguinal lymph node. This is a movie of bacteria trafficking in a lymph node after injection, targeting a tumor. This tumor has spontaneously metastasized to a lymph node and without treatment grows very rapidly. However, when the lymph node metastasis is treated with the S-typhimurium A1R mutant, the metastasis regresses and eventually is cured. This is compared with the untreated lymph node metastasis, as we saw previously, rapidly grows. We see from these data that the bacterial treatment did not affect the body weight of the animal, indicating lack of toxicity. The bacterial treatment also did not affect the primary tumor size, in contrast to its effect on metastasis, as we've seen before. The next series of experiments are the targeting and killing of lung metastasis of a human sarcoma in a mouse model by S-typhimurium A1R. Tumor cells were injected IV, and then 4 days later, bacteria, the A1R, was injected IV as well. The bacteria were injected again on day 11, and on day 16, we performed imaging to see the effect of bacteria on the tumor cells which had seeded the lung after the initial injection through the tail vein. The upper micrograph shows the tumor cells seeding on the lung before bacteria injection. The lower micrograph shows the bacteria targeting the lung 1 day after IV injection. We can see here with the excised lung that the treated animals that were treated with the A1R bacterial mutant, had no seeded cancer cells. This is in contrast to the excised lung from the animals that were not treated, where numerous colonies of tumor cells expressing RFP can be seen. The quantitative data show a very large difference in the number of lung metastases between the treated and untreated animals. The next series of experiments are the treatment of bone cancer and subsequent lung metastasis with S-typhimurium A1R. The human osteosarcoma cell line 143B-R was transplanted to the leg bone of mice. On days 7, 14, and 21 after tumor implantation, the animals were treated with S-typhimurium A1R. On day 28, the animals were imaged to determine the efficacy of the treatment. The untreated group had numerous lung metastases, as can be seen in A and B. In contrast, the animals treated with S-typhimurium A1R had no detectable lung metastasis. Quantitative data show that the animals treated with S-typhimurium A1R had almost no detectable lung metastases, as opposed to the animals that were not treated that had numerous lung metastases. The next experiment will be on the safety of S-typhimurium A1R. After IV injection in a non-tumor-bearing nude mouse, A1R will grow to a certain extent in the liver. However, by day 14, the infection is cleared. After intravenous injection in a non-tumor-bearing nude mouse, A1R will grow to a certain extent in the spleen and then be cleared by day 15. When the animals are treated with A1R, their survival is 100% over a period of 20 days. When wild-type Salmonella&typhimurium is injected into the mice, they're all dead by day 2. Conclusion: we could see no obvious toxic effects on the host treated with S. typhimurium A1R. Our data indicate that bacterial treatment of cancer, even without chemotherapy, is a very powerful approach. We are looking forward in the near future to test this powerful treatment modality in human patients.